3 labs: acids, bases, and buffers, pipettes, serial dilutions, and aseptic technique. These are really going to help you be prepared to do all of the microbial work the rest of the semester. I'm also putting a really big emphasis on what things look like and how we actually do them in real life, so that you'll be prepared to go into a lab at some point when the pandemic is over. Okay, so every week I try to give you a study tip. This week I'm going to tell you that repetition is key. So the first time you are in a lecture or listening to a lecture when it's distance education, you should pay close attention and take your notes, okay? But after that, you should just listen to it while you're doing something else. So if you were in person, you would ask permission to record the lecture, and then you would replay it as you're driving home, as you're doing other things. For this class, you already have everything recorded, so the very first time, pay close attention, take your notes, and then you can re-listen just to get that repetition, to memorize, do the first level of learning as memorization, and then you could do that during exercise, cleaning, whatever you want. Okay. You can also record yourself, and that's very, very useful for lots of reasons. So I really encourage you to practice recording yourself, explaining terms and concepts. Just being able to do that part alone is going to show you how much you understand and remember. But then you could also play back and hear yourself defining things as well. Okay, so we have a lot to go over, so let's go ahead and jump into it. Okay, safety is very important. Always look at the symbols, look at the instructions, and follow them. This applies to even things you're doing at home. There are lots of home cleaners and things that can be very dangerous. Acids and bases are corrosive to metals, and they're also corrosive to your skin. So you always need to protect your skin by wearing gloves, protect your eyes by wearing goggles, protect your airways by not working with things that are more concentrated than you are supposed to, and working in an area with lots of airflow. And in the lab, that might even include working inside of a safety cabinet, fume hood, okay, to protect yourself. Okay, PPE, personal protective equipment, even when you're at home. Biohazard is something you'll see a lot in microbiology. It only applies to biohazardous materials, microorganisms, right? Radioactive, I doubt you'll see, but if you work in a hospital, you will see that. Never expose yourself to radiation unless you have to. The rest of these, I think, are self-explanatory. Always make sure you are working with things safely. Okay, so you already know you can find acids and bases in your home. And sometimes you can find things in your foods, especially acids. Industrial processes tend to have very strong acids, bases, and other chemicals. So if you ever work in an industrial setting, you really have to know what you're working with and how to do that safely and how to dispose of things properly and safely. Furthermore, for our applications in our class, we care about the body and how the body functions, and we have very specific pHs in the body, like your blood. Down here, I have defined it for you, pH 7.35 to 7.45. Anything below this would be too acidic, anything above it would be too alkaline. And why is it such a strict range? Because of the enzymes in your blood and the proteins in your blood. We need everything to stay folded correctly. We need everything to function and work, okay? Enzymes in your stomach, on the other hand, would need to have an optimal pH of, 1 or 2, something very acidic. All right, so you are familiar with the pH scale. You know that it runs from pH 0 up to 14. Why? Well, these exponents, okay, the concentration of your proton and the concentration of your hydroxide ion will always add up to 10 to the negative 14. So notice here, when your protons are very scarce, when you have 10 to the negative 14 protons, your hydroxide ions are all the way at 1, as much as you can have, right? And so this scale, you'll notice, let me get a pen to write, and let me change the pen color to blue because we don't like red. So notice that your hydroxide OH minus ion, when you go from 14 to 0, you're going less and less, right? The concentration of OH. As you go from a pH 0 to a pH 14, your hydroxide ion, I'm sorry, not hydroxide, H plus, becomes less and less to 10 to the negative 14. So it's really important that you memorize this. But to memorize this, you really just need to know 0, right? Both sides of 0, the direction that it's going, and then you can recreate this whole chart pretty easily. Another relationship I would like to point out to you, little quick trick. If your pH is 0, then your exponent is 0. If your pH is 1, your exponent is -1. 2, -2, 3, -3, and so on and so forth. So you know, I could give you the concentration of protons, and you could tell me the pH, and you could tell me the concentration of hydroxide ions, and vice versa. I could give you the pH, and you can tell me my ion concentrations, okay? Because they are all connected, interrelated. They have this nice relationship. Another thing, very important, the difference between 0 and 1 is not 1, it's 10. Okay, so there are 10 times more hydroxide ions from 0 to 1 pH. There are 10 times fewer H plus ions from pH 0 to 1. Okay, so each whole pH value is 10 times less acidic than the previous value as you go from 0 to 14. Okay, very important. So the pH is telling us about the concentration of ions in solution. It's telling you how a substance behaves with water. Okay, and that we'll see on the next slide. So notice pH 7, you already know, you've memorized, is neutral, right? pH 7's neutral pH, but that is not 0, okay? People tend to think, they confuse this. If I asked how many ions of, H plus, how many ions of OH minus, it'd be easy for you to accidentally tell me 0 because it's not acidic and it's not alkaline. But it actually just means that they're at equilibrium. And so they're equal to each other. So -7 and -7. Okay? So if I asked you pH 4, what's the difference between pH 4 and pH 5? You would tell me that there's 10 times more hydroxide OH minus ions and 10 times fewer protons H plus. Okay? Okay. Now, I told you it's about water, right? We always say in solution, and it's because we want to know what's happening to our water molecule. Water itself is slightly reactive due to this attraction. You already know about hydrogen bonds. Every once in a while, this hydrogen bond actually, you'll actually see the proton, the hydrogen, go onto the other. Instead of just having the hydrogen bond with the attraction, every so often you'll actually make a new molecule, which you see here. We've abbreviated it as H plus, but really it's the hydronium ion, H3O plus, right? We took a water molecule and added an H plus to it. So if you have a substance that you add to water, if you have an acid that you add to water, first it releases the proton, but then the proton's going to end up adding onto a water and forming a hydronium ion. So we say H+, and that's totally fine, but it's really hydronium. And so when we look at the equation for pH, you could say H+ like we do here. It's quicker, looks nicer, but you could also substitute and put in H3O+, and that's the same thing, essentially. Okay? Now, I already talked to you about this equilibrium. When you're at a pH 7, you are at equilibrium. And that's because, remember, we looked over here at the scale, and the total sum of H+ and OH- is always going to give you 10 to the negative 14. And so if these are at equilibrium, we know that together they're equaling 10 to negative 14. And if you want to take an exponent and bring it down, you take the log, right? And so if you take the log of this number, right? So first we want to get one of these by themselves. So we get H+ by itself. So I didn't really show this step, so it's maybe a little confusing. But you have H+ equal to 10 to the negative 14 divided by OH-, right? And you do that on each side, and you eventually find that H+ will be equal to 10 to the negative 7, and you plug that into your equation for pH, and you get pH 7. And so that equation you'll see on the next slide. All right, so what is the definition of an acid, right? It is a substance that releases a proton in solution, H+. And of course, we know then that H+ is going to combine with water to form your hydronium ion, H3O+. And the same for a base in the opposite direction. It's going to steal a proton from water, and it's going to form your hydroxide ion, OH-. So a base is a substance, we say, that can accept a proton, but really it's stealing a proton from water, okay? And I already told you, acids and bases, they corrode metals and they burn your skin. And what we saw with this, water can be made into hydronium and water can be made into hydroxide, so we call it amphoteric. It can donate or receive. This is the pH equation, which we kind of already worked with indirectly. I didn't write it out, but we knew that if we wanted to bring something up from the bottom of the equation, 1 over H+ is going to be equal to the negative log of H+. Okay, and so you'll use this equation a lot. You'll also use this equation sometimes when you're working with a base. So we learned that pH 0 or pH 14, hold on, let's go back. It's always good to have a visual. We learned that pH 14, the H+ is 10 to negative 14, right? And hydroxide is 1. And on the opposite end, pH 0, you have 1 concentration of H+ and negative 14 OH-. And so if you think about that, and you relate that to the pH, or you can use the equation to solve for it, okay? But in either way, you'll see that, okay, if you're starting with pH 14 and you subtract the amount of hydroxide, that will give you pH. So pH is looking at H+. If you somehow are if you're working with a base and you only have information about the base, the hydroxide ions, then you'll know to subtract it from 14, and that will give you pH, which is telling you about protons. Okay, so question for you. If you had a substance with a proton concentration of 10 to the negative 6, what is the ion concentration of hydroxide ions? So if you have H plus, right, concentration equal to 10 to the negative 6, what would be your concentration of hydroxide ions? Well, you know that it totals 10 to the negative 14. So simply subtract 6 from 14. So what is 14 subtract 6? It's going to be 8, right? And so then we know, oh, so our OH minus is going to be 10 to the negative 8. If the proton is 10 to the negative 6, the hydroxide is 10 to the negative 8. Okay? This is really important that you realize we are working with strong acids and strong bases in this lab. If you were to work with weak acids, you would have to do more steps because you need to know how much actually dissociates. So strong acids and bases completely dissociate in solution, meaning that you get every single molecule of the acid will break up into H plus and whatever its conjugate base is, okay? And same for the base. Completely dissociates. And then all of those, looking at the acid, all those H pluses will then go and form your hydronium, and that's how you get your pH. If it was a weak acid, only some proportion of the substance dissociates, and you would need to know that number so that you could find out the pH that way. So we're not going to work with weak acids and weak bases, but it's not that much harder. You just need to know that the dissociation constant for that particular acid or base, and then the rest of it's the same. Okay. So calculating pH for us, we're working with strong acids and strong bases, so we know it completely dissociates. So the concentration of your hydrochloric acid solution is simply equal to the concentration of hydronium ions that you're going to form. And so you can simply take that concentration and plug it into the pH equation. And remember, pH is equal to the negative log of H plus, okay? And we just said here that HCl is equal to H plus. And so you plug it in and you get this number, 2.9. Nice and easy, one step. We like that, right? If it's a base and you're asked to find the pH, remember that pH is telling you about H plus. So to go from OH to H, you need to do that equation. pH is equal to 14 minus your OH concentration. And so that's what we see here. So we take a strong base, it completely dissociates, so the concentration of that base is going to be equal to the concentration of hydroxide ions that form. So we take that and we plug it into this equation for OH, which is POH is equal to the negative log of OH minus. Since I'm working with the same number that I did in this example, I still get 2.9, but that's telling me about OH, right? So we want to get H because the question asked us what is the pH. So we use this equation, pH is equal to 14 minus POH, which is what we just did up here. So we plug that into the equation, we subtract it from 14, and we get a pH of 11.1. So very alkaline, right? Just like this was very acidic. Okay, so I think you have all of the concepts at this point for working with acids, bases. I have some practice questions for you to do on your own, which you'll see in a little bit. One more concept about acids and bases is then working with buffers. Okay. I ran out of room on the previous slide to show you this chart, but this is really, we've already talked about this, right? We were talking about hydrochloric acid, we were talking about sodium hydroxide. So just to give you a reference of some of the common acids and bases that you'll see, here's this chart. But moving on to the last concepts of this lab 3, is salts. So what are salts? An acid plus a base will give you a salt. And remember, the acid is going to dissociate in solution. The base is going to dissociate in solution. And what happens is you get this rearrangement where the Cl minus is attracted to the NH plus in the solution, and they form a salt. Because this is hydrochloric acid and sodium hydroxide, the salt that forms is table salt or sodium chloride. If you're working with a different acid, a different base, you'd form a different type of salt. And you always also end up with water, because H plus and OH minus, when you put them together, you get water. Okay. Another thing about salts is that electrolytes in the body that conduct electricity, these are salts that, they dissolve in solution. The solution is our blood in this case. And so then now you have these ions in your blood and also in our cells. And it's the whole reason we're able to, our whole system, neurological system, is based on this electrical conductivity. So when you get really dehydrated, whether that be from being very sick or exercising very hard or being somewhere in a very hot day, not only do you need to replenish your water, but you also need to replenish your electrolytes. We tend to do that by having a sports drink. And you'll notice those sports drinks, they do have sodium, but they also have some other salts like magnesium, for example. Okay. Furthermore, the other reason we're talking about salts, not just because of electrolytes and not just because of neutralization reactions, but also because of buffers. So buffers are substances that resist a change in pH because they can either accept or donate depending on if you start to accumulate more of one or the other. And so this is super important. Our body uses buffers to maintain those compartments of the body that have different pHs. So buffers are molecules or salts that slow the change in pH by either accepting or releasing a proton. And a buffer is always going to be a weak acid and a weak base, so it does not completely dissociate. It will only partially, and it can go in either direction. Okay, and so the most common buffer system in our body is the bicarbonate buffer system in our blood. And so this is the equation, and you can watch this video that will show you step by step how you go in one direction under certain conditions, and you go in the other direction under other conditions, which is why you have these double arrows here. In the middle, you have carbonic acid, our weak acid, right? And on the right side of this equation, you have the bicarbonate ion, which is our weak base. Okay, so if you start to really increase the amount of carbon dioxide in the blood, it's going to combine and make more carbonic acid, and it's going to shift that whole equation to the right. Carbonic acid, the more and more of it you have, you'll get more dissociation happening, and you will increase your protons. So, but more importantly, you're going to increase your bicarbonate ion. if you took physiology, you learned the specifics of how this is actually done in the body, right? We regulate the amount of this bicarbonate, and we regulate the amount of protons and the amount of carbon dioxide. So this is a relationship between your kidneys and your lungs. To get rid of CO2, you exhale it. But if your lungs are not working correctly, then your kidneys can step in and start to do some regulation. Again, because you always need to maintain your blood pH. If your blood pH gets out of range, you will start to have symptoms from that. You will have side effects from that. Okay, so bicarbonate will accept, this bicarbonate ion will accept protons if they accumulate, if the pH starts to fall. This will, so looking at it in the other direction, this direction, if your bicarbonate ion is accepting protons, it's going to form more carbonic acid in this direction, and then that's going to cause more CO2 to be released, and you'll need to exhale that out. Okay, that's assuming that your lungs are working and you can exhale the CO2. Okay. Again, you can watch this video for a little bit more visual explanation of this buffer system. Okay, serial dilutions. Serial, doing the same thing over and over again. I like to do 1 to 10 dilutions. 1 to 10, you can write it in ratio form, right? But you can also write it as a fraction. And then you can also write it as a decimal, right? And if you could take your calculator and say 1 divided by 10, and you'll get 0.1. Okay, so I've shown that here. But something I want to point out to you, because this is really easy to skip over. When you're doing a 1 in 10 dilution, you're going to put 1 ml of your stock or starting solution into 9 mLs, because 1 plus 9 gives you 10. So a 1 to 10 dilution means 1 into a total of 10. And how do you add 1 and have a total of 10? It's by starting with 9, right? So 1 into 9 gives you a 1 to 10 dilution. So this is our first 1 to 10 dilution. Doing it again, the total dilution would be 1 to 100, but you could also say 1/10 times 1/10, right? And the other way to write it would be, we diluted it, so this is now 0.01, right? So 0.1, 0.01, 0.001, and you can keep doing that, and you'll keep moving over 1 decimal place. That's why I like to do dilutions as 1 to 10. This is the most common, it's the easiest way to work. So I have a sample question here, okay? Your stock solution is 1 molar. You have 10 mils of it. You need to have a 1 millimolar solution. How do you do that? So you're going to put your initial concentration, which is 1 molar, and the final concentration that you want. When you're doing equations, you need to remember to keep everything in the same units. I've chosen to work with everything being molar. You could just as well change everything to being millimolar, but this is the way that makes sense to me. So if I want to change 1 millimolar into molar, milli is 10 to the negative 3. I assume you have your metric system memorized, but you could always look that up if you don't remember. And so 10 to the negative 3 molar, and again, you can always write that exponent out. So this is the concentration that we want. And this is the concentration, oops, didn't mean to cross that out, but this is the concentration we're starting with. So how do we get from 1 molar to 0.001 molar? We dilute it using serial dilutions of 1 to 10. So what does that look like? Our initial concentration of 1 divided by our final desired concentration of 0.001, that gives me a difference of, it gives me a dilution factor of 1,000. So I'm going to dilute this 1,000 times, but I'm not going to do it in 1,000 steps. I'm going to do it in 1 to 10 dilution steps. So how many times does 10 fit into 1,000? Divide that out and you get 3. So I just need to do 3 serial dilutions. So 1 to 10 dilutions. And again, remember that's 1 mil into 9 mils to give you a total of 10 mils for your serial dilution. So this is the most important. These are the important concepts to learn for serial dilutions. And in real life, this is the most common type of serial dilution, is a 1 to 10 dilution. However, your simulation likes to do 1 to 5 dilutions. So I want to review that with you. 1 to 5 means 1 part of substance A and 4 parts of substance B for a total of 5 parts. Okay, so now let's apply this to a scenario where we want a total volume of 100 microliters, and we know that there have to be 5 parts, equal parts. So take 100, divide it by 5, and you get 20. And then remember your units, it's microliters, okay? So I know I have 5 parts of 20 microliters, and we want 1 part of one substance, and we want 4 parts of another substance. So we know that we're going to have 20 microliters of substance A and 4 parts, 20 times 4. So that gives me 80 microliters of B. And then you check your work by adding those together, and you do in fact get your desired volume of 100. So this is a 1 to 5 dilution. Again, I had to have been told originally that I wanted 100 microliters. If you're not told a volume, you're just told make any 1 to 5 dilution, you could pick any number you want. 100 is an easy number to work with. That's why I chose it for this example. Okay. In that pipetting simulation, you will also see our pipette tips I have hiccups. Our pipette tips have to be sterilized. And when we sterilize materials in the micro lab, is most often done using an autoclave, as long as the substance is able to handle the high heat and high pressure of an autoclave. Now we want to know which items have been autoclaved and which ones have not. Things can get hectic in the lab. Furthermore, we want to know that the machine actually worked and did its job. So we use this autoclave tape. It's just looks like regular tape before, and then after it's been autoclaved, as if that temperature was reached, if the sterilization actually happens, it turns the tape striped. And so you know anything with the striped tape has already been autoclaved. And so that's really mainly what I wanted to show you. But I also want to point out that the reason we use an autoclave is because it's basically a pressure cooker. We are able to use steam to build up high pressure and get to a very high temperature, which kills everything, even the most resistant bacteria, even endospores are killed by autoclaving for, now we say 15 to 20 minutes, but that's at the high heat, high pressure. So it usually takes Depending on the machine and how good it is, it can take 20 to 30 minutes for it to get up to that temperature. Take another 15 to 20 minutes for the actual sterilization, and then there's going to be time for it to cool down to vent off the steam, and that can take anywhere from another 10 to 30 minutes. So the entire autoclave cycle could take a couple of hours. I think the fastest I've ever seen is about an hour and 10 minutes. Like I said, it just depends on how good of a machine you have. It's going to take time to reach this temperature, then you have to maintain it for this time, and then it's going to take time to cool off and be safe to open up again. So that's enough. We will have an entire lab devoted to sterilization techniques and disinfection techniques. But for now, for an introduction to how to work with microbes, this is good enough. And this is just a diagram of what I just said about how an autoclave works to put in steam, build up that high heat, high pressure. A couple things I want to note about how to use the autoclave safely. In addition to using the autoclave tape, you also want to make sure that you never have a sealed container. It can have a very loose fitting cap, as long as it's not, if it's a screw top cap and you put it on like very, very loosely, that's okay. Or you could cover it with foil. And that's about it. Anything else like a cork or any other type of lid, the pressure would pull the lid shut and then you would have a closed container. So you just have to be mindful of how you're doing it. Also want to make sure, you would never fill something that full. It would just boil over. And then you want to make sure that the material is safe for the autoclave. So things like antibiotics are never put in an autoclave. They would be broken apart. They wouldn't function anymore. You would ruin it. So instead we do what we call filter sterilization, which you'll see later. Okay, so pipetting. Now we've done all the work to get our sterile pipette tips. We need to make sure that we have a biohazardous waste container to put or dispose of the used tips. You always use a fresh tip for every transaction. So in between, even when you're doing those serial dilutions, each dilution gets a new tip. Okay, each 1 to 10 dilution gets a new tip. And then making sure you have the correct pipette tip size for the pipetter that you're using. Now I'm saying pipette and pipetter because I'm lazy. It's really a micro pipette, micro pipetter, micro pipette tips. Okay, because these are all small sizes. The largest size that you're working with is one we call a P1000, which means it can go up to 1,000 microliters. 1,000 microliters is equal to 1 mil. So very tiny volume, and that's the large volume, and it gets smaller after that. Okay, you definitely need to know these sizes, what the lowest volume you can pull up and the largest volume you can pull up with that pipette size. Okay, so now you've done all that, you need to know how to correctly hold the pipette and how to use it to pull up a solution, and how to use it to release the desired amount. Okay, so there's a dial on the pipette, which is not shown here, but click on this link and watch real pipettes being used. Okay, and so you would set the pipette to the volume that you want, and you would hold the pipette so that your thumb is on the trigger. You're going to slowly press your thumb on that trigger to the first stop, which you'll feel that you would have to apply even more pressure to go past it. So you'll know that that's the stopping point as long as you're going slowly. You would also do that before you insert your tip into the solution. Otherwise, you're going to blow air bubbles and possibly displace your solution, and that's not good. Okay, so you always press down slowly to the first stop, releasing that air from the tip. Then, keeping your thumb on the plunger, you will stick the tip into the solution. You want to go middle or a little bit towards the bottom. You do not want to actually touch the bottom because then you'll block the flow. Okay, and then once your tip is here, you can slowly release your thumb, which will draw solution up and into the tip. If you don't do it slowly, you will get air bubbles, and then you don't have the actual amount that you were trying to get. When you go to transfer that into a new container, you do so at an angle, not necessarily so high, maybe down here, and you slowly push down again to the first stop to release that fluid into the tube. You don't want to go straight on an empty container, and you don't want to go fast again because it can splash out, and then you have to start all over, and you might only have one sample. So you need to be mindful of these correct methods. Okay, if you had a container that already had some fluid in it, then you could stick your tip into that solution and slowly push down to release solution that way, and that's okay. But really, you can't go wrong if you use this angled method along the wall, slowly drip it down. It takes so much longer to explain how to do it than to actually do it. Okay. Please carefully watch this. This will show you what it actually looks like, how to correctly do it. And I really want to point this out, that your simulation is going to confuse you because it's going to ask you about like how to get all of the solution out of the tip. And the correct answer to get all of the solution out of your pipette tip is to push all the way down to the second hard stop. But you would never actually do that in the lab because that would be more than the amount that you're trying to measure. So it's a weird, tricky question that's not helpful. So I really wanted to point that out to you, okay? And then the last thing about the pipette simulation is that to give you something to actually do with pipetting, they're having you do this Bradford assay, which is to measure how much protein you have in a solution. So we use a dye that we know will bind to protein, and we also know the wavelength that we can measure this dye using a spectrometer, and we measure the absorption rate at this wavelength for this particular dye, and we can extrapolate the amount of protein we say is equal. The absorption is proportional to the amount of dye that is bound to protein, and hence, therefore that's going to tell you the amount of protein that you have. So the amount of dye you have is the amount of protein you have. The only problem is it's really sensitive. The spectrometer cannot work with super concentrated solution. So we have to dilute our sample of protein down to a very small amount, maximum half a milligram per mil. So that's why you had to learn how to do dilutions. And then you can dilute it, add your dye, and then make your measurements. And from this, you'll find out your protein concentration. So that's the Bradford assay. And I can't remember, but I think you might see this once more in our Labster series. So, Pay attention. Okay, so here are your practice questions. I'm not going to read these to you. You can look at them on your own. Take time, think about them, really try, and then confirm your answers in office hours. And that will mean that you understand what you need to know for working with acids and bases and buffers. And then lastly, we're going to go over aseptic technique real briefly. And I have questions for that as well for you to practice. Plus your actual post-lab discussion questions. So you have more than enough to master these topics and be prepared for your exam. Okay, so aseptic. A means without, septic means contamination. So without contamination, we have techniques to prevent contamination. We want to protect the integrity of our lab work, so we know that we only have one type of microorganism that we're working with, and therefore the results that we get are because of that one microorganism. If you have more than one in your solution or on your Petri plate, you won't know which one did what. You don't know. Okay, so that's the first reason we have these techniques. The next reason is that we don't want to get contamination on the lab area, the lab equipment, and we don't want to get it on ourselves, right? We want to protect ourselves, we want to protect our working environment, and we want to protect our the integrity of our lab work, okay? So to do this, we use a Bunsen burner. We use the Bunsen burner to create a zone of, I don't want to say sterile zone, but it is pretty much a sterile zone around the Bunsen burner where you can safely do your work and greatly reduce the risk of having microbes just fall from the air into your what you're working with. Of course, you still need to be careful, and you never get super close so that you don't breathe on your, you don't want to breathe onto your work, because then if you have microbes that you're exhaling, they would just get into your work, right? You also don't want to breathe in the microbes that you're working with. And then, so you're going to watch this video. It's going to show you step by step how to work with a Bunsen burner, how to inoculate liquid media, a solid media like a Petri plate, and another solid media, which is called stab media. It's going to show you how to work with a liquid culture and a solid culture to do these inoculations. So inoculate is to introduce your bacteria into a sterile media so that you can start growing more of that particular organism. And so remember that microbiology is a very detailed work. So you need to pay close attention to the steps. How are you holding the items? The angle you're holding them at, and the order that you do things in, that all matter. It's very detailed. So pay close attention, okay? At the end of that video, you should be able to answer all of these questions. Read them before you watch so you know exactly, what you're looking for. You might want to watch that video a few times. And then once you're able to answer these questions, you will be able to do your simulation pretty easily, and you'll be able to do your post lab pretty easily. Okay, so you're all set to do all 3 labs. And then come to office hours and confirm your answers, anything that you're not sure of. All right, good job. Bye.